Hello, everyone. Welcome to Hackathon for Neurofibromatosis. Thank you so much for having us here. Uh, we have made simulated R2D2 cell lines for drug screening, along with my teammate Dhruv. Didi. Now, coming to the problem, we know neurofibromatosis is a multi-system disorder, and if you understand by MPNST, it's aggressive, needs chemo. Clinical trials are ineffective as they predict only few drugs, and in silico analysis does not yield drug response. So we need cell lines, but the cell lines are hard to get because it takes time, it takes resources, it takes trained people, and it takes a lot of time, which might make the cancer patient die before we intervene, and translation is missing. Now, so for solution, we propose this in which we, just like in vivo, we see gene expression, and just like in vivo, from proteins we have particular pathways. We did gene set enrichment analysis and analyzed pathways. Just like in vivo, a drug interacts according to pharmacogenomics. So we uh, gave drug interaction score, and we received also drug gene interaction from drug gene interaction database, and we trained a model which is dynamic. So presenting to you simulated R2D2 cell lines for drug screening. Begin with the first step was to process the data, where we did differential gene expression. Then second was to find a correlation between genes and pathways, for which we did gene set enrichment using reactome.org, and then we calculated something unique to our solution, which is gene frequency. Then we devised this gene drug relevance score. Upon the basis was gene druggability and drug gene interaction score. And finally, we got the results in which we have very well predicted the response for 5-fluorouracil, methotrexate, cyclophosphamide, and epirubicin, according to the pre-existing cell line data for drug screening that was given to us, and also nilotinib. Now, passing to Dhruv.
Yeah, I think this was a great opportunity for me as an engineer to work with Arva on this. And we're really happy about how we can use this technology, put it off to clinics, and essentially use the correlation between pathways, genes, and drugs that we've created here to create predictions for all kinds of treatments. Thank you.
Now, Drew will show us in real time the HTML profile where we can see the matrix.
So if you, in this, we are able to see how different values we get from different matrices across.
Sharing the screen.
Yeah. And here is the code which was used to generate this. Back to Arva.
Our solution provides us a link between omics, AI, and also the drug gene interaction, and provides everything directly to the clinic. So we bring translation to it, and we hope that it brings solutions to neurofibromatosis problem, as we are lacking data. For a lot of cancers, we are lacking data. So if any questions are there, we would like you to address to our respective email addresses. And I'm very glad and feel fortunate to be a part of this hackathon. Thank you so much.